Welcome back. This is the last segment on mutualisms, certainly on mycorrhizae. I am sure I'll bring it up again in other segments, but this is the last segment where I'm going to talk about the mycorrhizae, and I'm specifically digging into my research in some cases here on ectomycorrhizae. Very important group. As I ended with the last one, 20% of the world's plant species are associated with ectomycorrhizal fungi, and you could review what plants we're talking about. But just remember all of the pines and all of the oaks and beeches and such. A lot of very important trees in our Adirondack forests. So here are some images. You've seen that slide with the hyphae around the roots of the pine. Again, that's a swillus on a pine. On the left are a couple other mycorrhizal types that have been nicely cleaned up by Zach back in the day. And panel D is a fungus you know. So those are different plant species. Panel D is on Picea. You wouldn't be able to know necessarily. It's a white hyphae on Picea. So the root tips are coated or wrapped in a sheath of hyphae. The plant root is dark. You can see that here, right? You can see it over here on this panel. That's the plant root, okay? But then the root tips are wrapped by a blanket of fungal hyphae. Up here, it's the same story, but this particular species of hyphae makes a nice, wefty, hyphal, cottony mass. The root tips are all back in here. The fungus is out getting nutrients with this wefty mass out here. This one has very well-defined rhizomorphs in panel B, formed by Cortinarius, if you know your fungi. So these are the channel of nutrient water flow between fungus in the soil and root tips. This is formed by Cortinarius. And then this one back to panel D. This one's formed by red cap with white dots. You've seen it on salt shakers. You've seen it in cartoons. Maybe the Mario Brothers, or is that just a russula, red without the white dots? This is Amanita muscaria. I'll show you a picture of it later. Okay, so there's a lot of morphology. It's a very interesting group, and what I've been doing for my research is pulling them out, cleaning them up. I don't know who it is normally, just by the morphology, but I can extract DNA and then identify what fungus it is with the DNA, even if I don't have the mushroom around to link it to those root tips. Here's a picture of a Lacaria mushroom associated with a pine. Okay? And you can see down in the soil pot here, the roots are long and running down to the bottom, and then there's a branching of the roots of the plant, kind of an amber color, almost the color of the Lacaria, but that's an unfortunate coincidence. The root tips are the color of the hyphae that the fungus has wrapped around the plant roots. And so I'm going to blow that up. Now you can see, here's the plant root in its amber color, brownish, and then all the root tips here are whitish because of the fungal hyphae wrapped around those root tips. Those root tips would typically have root hairs, but they don't now because they have all that fungal hyphae. And you can see some of the fungal hyphae out in the soil here. So the fungal hyphae leaves the root tips after it gets all that sugar and grows out into the soil to get more nutrients for the plant and itself, right? Same story, for itself. And then if you do a cross-section of one of these, like you're slicing a carrot into those discs, you could look at the fungal interaction. Now this is a different fungus. It wasn't white, it's brown, Thalephora species, but that doesn't matter. And that's the mantle. So the fungus is bringing nutrients back and it stores it in that mantle, nitrogen, phosphorus, calcium, etc. And then occasionally delivers some into its hyphae that go into the root of the plant. These do not go into the root cells. Remember the arbuscular type filled those cells. These don't do that. They stay out. They don't cross that cell wall. They form this network of hyphae, and that's where the nutrient exchange occurs in a conifer, in an oak, in an alder, etc. The plant is sending sugar down here. That gets out into these cells, the cortex cells, and then that gets taken up by the plant, the fungus, excuse me. And then the fungus again is delivering water, nitrogen, phosphorus, etc. This region, this network of hyphae honors the guy that used to think they were all pathogens. It's called the Hartig net. He was that forest pathologist that said all those fungi are pathogens. But Franck decided to give him the honorific title and say, oh, we'll call this thing the Hartig net. That's what we call it now, just for fun. Here's one of Franck's etchings. He had a horrible microscope in 1890. You who complain about microscopes in our labs, this guy had it worse, and yet look what he was able to do. You could see he even had the Hartig net very well. So these hyphae are on the top side of those cells. These hyphae are in between the cells. And then this is the mantle. This is the mantle. This is extra hyphae out into the soil and the rhizomorphs, etc. So he did pretty well with those old microscopes. He was, by the way, I think I already said it, he was a lichenologist, so it was not out of the question for him to kind of have a feeling that fungi were not necessarily all bad. Here are some species of fungi. There's our Amanita muscaria. I promised I'd show you a picture. A couple of truffles. This is the Oregon white truffle. This is a basidiomycete truffle, rhizopogon. Fun fact, I was involved on a paper. I'm not allowed to do it, or I chose not to do it, but I was involved in a paper where we named one of these fungi after my wife. More on that if you're interested. Chanterelles, delicious edible, right? So are those tuber Oregon white truffles. They are very common in the forest floor. Right now, chanterelles are growing in our area. But I could tell you that now because it's much later when you're watching this, so you're not going to go out and pick all my chanterelles. Okay. By the way, some of these don't produce stems, caps, or anything. They don't make a spongy layer. They don't make gills. They just make spores in the litter. And they're very important on our root tips. Often the most abundant type on the root tips. You never see it. You walk right over it because it's just a smear on litter layer. This is the real reason why I love these. They are delicious edibles. This is, again, the Oregon white and Oregon black truffle. Porcini from one of my field sites. This is a normal toaster oven. It's not a dollhouse. Those are huge, and they were delicious, and no maggots. I don't know if you can tell, but there's no maggot holes down there. If you know your fungi as edibles, you always have to worry about maggots. These are matsutake. That's been driving a lot of the economy in the Pacific Northwest for quite a while, since forestry got curtailed for a while in the 80s. Matsutake, it's a delicious fungus, and the Japanese treat it like we treat truffles. These are my hands. That's my basket. Beautiful porcini harvest somewhere in New York. I will not tell you where. So delicious edibles right across the board. If you're interested in learning more about that, join a club. Don't go off with a field guide because you'll make a mistake and it can be deadly. But if you get that TEK, traditional ecological knowledge, from a club member who says, I've eaten this fungus and I'm still alive, you're much more likely to have a good experience than a bad. If you're trying to figure out if you've got the right thing with a field guide, there's far too many species out there for field guides to have everything. And so you may get yourself into some serious trouble. Don't do it. So here is a little bit on ecosystem level interactions. And this is largely why I wind up becoming a mycorrhizologist in the end, because money was coming in to understand the fungi because of this guy. I think that's a guy, could be a gal. The northern spotted owl was known to have, at the time, I think it's been challenged a little bit, but have old growth specificity. And so environmentalists were able to stop logging operations in old growth forests because they would find the northern spotted owl and be able to say that's an endangered species and we need to protect the forest for this owl. Why did the owl need the old growth? He wasn't logging. He was eating these guys, the flying squirrel. And the flying squirrel would basically glide down from the treetops down to the ground and dig up truffles. and eat them. And 90% of the flying squirrel diet during the fruiting season of the mushrooms is truffles, native truffles out in those forests. And it's dispersing the spores in its fecal pellets. I've got, my students and I have shown that in some very interesting successional patterns. So it is a symbiosis in a sense. But importantly here, if you don't have the truffles, you don't have the flying squirrel, and you won't have that northern spotted owl, at least as abundantly as you would if you do have them in that forest. And also they're helping the trees. The forest is benefiting from the mycorrhizal fungi. So those squirrels, those fungi, those owls, those trees are all interacting in an ecosystem level. And this has been shown also in a paper that Suzanne Simard, and you'll see her name in a minute, published in '97. I was looking at interactions between different hosts and how their fungi might colonize the roots of both plants. And then we could say, boy, there's something going on below ground that we don't account for in our competition theories about plant light competition. And it turns out the fungi are connecting these, at least genetically. I could show the same species were on the different tree hosts. And that blows competition out because the fungi are interacting with both hosts and want both hosts around necessarily. What Suzanne did is labeled this host, when it was green, with an isotope of carbon and saw that it showed up in this host. And so, and presumably through the fungal networks. And this was it was a great story. It was right around when www was the hot thing, the World Wide Web, and the Nature. These are the guys that published the double helix. This is a big journal. They came up with this cover item and called it the Wood Wide Web, kind of a, I don't know, propaganda. Here are her data out of that paper. So she shaded Douglas fir either with deep shade, partial shade, or full light. Okay, and then she added labeled carbon, C13 instead of C12, to the leaves of, in CO2, to the leaves of the poplar, or aspen, I believe. And then she dug up, well, then she harvested leaves of the Douglas fir and looked for the labeled C13 in those leaves. And so in year one, there wasn't a whole lot of label anywhere, whether the Doug fir was growing in a shaded environment, partial shade, or full light. By the way, these are little field microcosms that were maybe a meter in diameter. And she had a control plant that was an arbuscular mycorrhizal plant that looked very much like the Douglas fir in year one. There wasn't a lot of transfer of anything to anybody. But after the fungal networks grew for a year, in year two, she redid it. And now she saw in the deep shade especially a huge spike in the labeled carbon coming from the other tree host through the fungal network and into the Douglas fir that was in the shade and was starved for sugar, wasn't able to photosynthesize enough. It even worked in full light. There was a little bit, not a huge amount, but a little bit. So pretty cool story, made a big splash. There's her name, Simard, S-I-M-A-R-D. She's got TED Talks, she's got articles, students. Look her up. She's got some pretty cool stuff out there. And others have looked at this and decided maybe something else is going on. They saw it, they showed with carbon-14 transfers between trout lily and maple saplings, that the carbon moves to the trout lily during part of the season and then it reverses to the seedlings later on, or vice versa. I forget how it works. But a nice little paper in Ecology by Lerat et al. From our northern neighbors here up in Canada. This happens. Whether it happens with green plants in an ecologically meaningful way is a bit of a controversy. It happens, but does it really matter? One could argue, I suppose, either way, and have. It's kind of a boring argument probably, but the fact is it can happen, and these plants have evolved a way to capitalize on that. They don't produce any green parts. They don't have any photosynthesis. They evolved out of the blueberry family, and they're very good at just putting up a flower without any chlorophyll. And the way they do that is they parasitize the fungi that are associated with the trees in the stand. So you know this one, some people are now calling this ghost pipe. They don't like the word Indian pipe. Ghost pipe, snow plants, these are out west, etc. I love these plants. I think they're awesome. They are true parasites, but not of the plant. They're not tapping, like a mistletoe taps into the phloem of the plant. These are tapping into the mycelial networks that are getting their energy, their sugar from the plant, and then the fungus, this plant, the snow plant, taps into that and steals that sugar. So here's a cartoon I did years ago. You're walking at Heiberg in the fall. You look around you, you know sugar is being delivered below ground. Maybe as much as 50% of the photosynthate goes below ground, and maybe as much as 15% of that goes to the mycorrhizal fungus associated with those trees. So that's down at the root tip. They produce hyphae. That hyphae goes out into the soil and associates with the root mass of this ghost pipe, which eventually sends up a flower. And we know these are parasites in part because they're highly specific. Most fungi will reject this plant, but it has learned to override the resistance in the russulas. And so whenever you see ghost pipe, you will probably see russulas fruiting nearby. Probably not as much as they could if their fitness wasn't being challenged by this parasite. Okay, just a few more slides here. Mycorrhizal fungi can facilitate plant invasions. This is a project I've been involved with for some time, starting with Dr. Dan Simberloff and his student Martin Nunez. And then Martin Nunez got his PhD, and he has a student, Nahuel Policelli. We just got another paper accepted on the same system. If you look back here in 2007, there's a plantation. This is in Chile, and a road right there, okay? And some of the ponderosa pine had made it out there and become established in 2007. Probably a lot more seedlings you don't see. 8 years later, 2015, that's the same view. Note the hill there, and it's loaded with the conifer. And this little bump right here is seen right here. And then this was closer to 2019, and it's basically the invasion is on. And conifers are the world's worst invasive species, plant species, but they're invasive in the southern hemisphere. They don't occur south of Nicaragua. I'm not too sure about the European end of things, Mediterranean. But in North America, South America, they stop in Nicaragua, except we have planted them in Chile, in all over South America. Ecuador, I've done work. Argentina, over in South Africa, Australia, New Zealand, and everywhere we've planted them, invariably, eventually, particular species like ponderosa pine and Douglas fir and others become really bad invasive. The cool thing that Jeremy showed, he was my PhD student, is that he collected the roots from these plants. Next to the plantation and in the plantation, there was a small group of fungi that came with them in the plantation setting. But as soon as you get out about 10 meters and beyond, it's one fungus, Swillus luteus. So this invasion cannot happen without a particular species. It's a co-invasion with the mycorrhizal fungus. I'll just stop there on that. And to wrap it up then, mutualisms can improve the acquisition of water, nutrients, places to live, whether you're a plant, a fungus, an insect, a moray eel, a shrimp, a nudibranch. Mutualisms can aid in defense. Remember, there is different kinds of mutualisms: trophic, defensive, dispersive, and the fourth one. Mutualisms can change when conditions change, so they can basically be plastic against changing environmental conditions. And they can facilitate pollination, seed and spore dispersal, plant establishment. I've made my career on how mycorrhizal fungi affect where plants can establish, both in natural succession but also invasive systems. And finally, they definitely affect whole communities, and that's part of the establishment. So that brings us to a close to the mycorrhizae. You just heard a lot more about mycorrhizal fungi than any other ecologist out there this year, or any other ecologist at another university, because I don't think there's any mycorrhizal people teaching ecology anywhere else in the world. So, lucky you. I hope you can utilize these ideas even in your backyard on mycorrhizae, understanding that soil has to be healthy for your plants to be healthy. So that's it for this segment. We'll see you next time. Cheers. Thank you so much.